Yeah, thanks for those of you guys who did the extra credit. Well, thanks to you guys. I mean, you guys get the benefits, so hopefully everybody took advantage of that last class. Did you guys find it helpful and useful to kind of learn some of the little details that maybe can be fixed up for the final exam? Okay, cool. It's also a really good strategy to learn to do on your own, right? So you don't need me to do it during class. You can do it every day or every time you do a study session or something for new material too. It's a really good way to do that. Draw out the material and label it so you can put the names to it. So that's one of the reasons that we gave that full PDF for free of the coloring book for all those kind of things to do the same process of drawing this stuff out, labeling it, really get to think and work with the material because it's a really good way to learn it. So, let's see here. As we get started today, were there any questions from where we left off with last class or with what you prepared for? Anything you came to mind? Yeah.
So there were studies about the rats with the doing the maze, I think it was, when they had their hippocampus and stuff cut out. And they said that they didn't know if it was because of if rats were any different than humans. Do they have any other studies, like more recent studies, that show that that instead of just the motor recall, I guess, that it does affect their overall memory, like their semantic memory and stuff like that?
Good question. So see if I understood everything. So you're saying, so you said it was cool that the studies they were talking about with the rats with the cutout hippocampus could do the maze as well, right? You remember the maze? Was it the Morris water maze? When you drop them in the water and they swim around? Try to find the...
I don't remember the exact...
Okay, yeah. So, and so you're asking, was there evidence that humans had similar issues?
No, it was, they determined that the rats did not lose any form of memory because of taking out the hippocampus, because they were only measuring motor skills rather than cognitive. Are there any research currently that shows that it does affect their memory cognition, if that makes sense?
There is actually, yeah. So that's some of the data that actually we talked about that last class, some of the work from Howard Eichenbaum's lab, and actually lots of other labs since those earlier days have done lots of maze and other studies with rodents, particularly chopping out the hippocampus and other parts of their respective medial temporal lobe structures, and find that there can definitely be some elements of memory that seem to be more cognitively related rather than just motor based. But it's tough. So you guys remember the conundrum I posed you guys last class about the rats? How do you ask a rat for its cognition? How do you know what's going on inside the rat's head? Yeah, you really can't, right? So the rat can't really tell you what's going on in its head. For those of you guys that came out to the Neuroscience on Tap conversation we had last week over beers at the campus pub, we had this example of the same thing. We said, how can you ask a rat its confidence, or how do you know what's going on in here? Because sometimes, even if you ask them, and ladies, correct me if I'm wrong, and you ask your guy, what are you thinking? Don't they usually just say, nothing? Most of the time it's true. That's really what's going on up here for us, nothing. Right? So even if you ask them, the rats might be like, no, nothing here. Just hanging out. So you can ask by looking at their behavior. And so that's what they did in some of the Eichenbaum work, and I'll see if I can maybe show some slides from that or some data points from that where you look at behavior instead. And so you devise ways that they can show you things, maybe through their motor activity. It's not motor that's impaired or preserved in this regard. It's actually, the motor is just a vehicle through which you can gain insight and understanding some cognitive elements of memory. So we talked last class about memory confidence, right? Which is one way in humans we've delineated between these two processes called recollection and familiarity. So does anybody remember what we talked about with recollection and familiarity last class? Just big picture, like what are they? How are they different?
For recollection, wouldn't there be a source of memory, whereas for familiarity, there is none?
Yeah, absolutely. Great job with that. So, good example there is when you recollect, it's tend to represent times when we can remember things really clearly with details and context. So, the source of the information. So, basically, if you're recollecting something, the idea is that you should be able to remember where the source of that information came from. So, did you hear this from me or Dr. Amadeo? You know, if you can't remember that, you just kind of know that, well, I'm pretty sure I heard this thing before. That's more of like when it's a familiarity type of recognition. And so we can look at that with memory-driven confidence, which brought us to this discussion about the rodents. And you can look at these cognitive processes of recollection and familiarity by virtue of looking at the motor behavior of where the rat runs to the cage, in the back of the cage, where there's a reward if it's a new item that they're seeing, or if they dig into a deep cup full of bedding that smells like an old odor that they remember, in which case their behavior is going to show you, I remember this from the past because I'm digging a lot to get this fruit, and I wouldn't dig unless I really did remember this clearly because there's a freebie at the other end of the hall if I'm wrong. See what I'm saying?
Yeah.
So really good question. I like how you're thinking about it. And yeah, and you're on the track because you're thinking about the way that lots of other people have, which is let's think of some ways to really look at some of the cognition. So, yeah, let me see if maybe I can find us a picture of. So if you need to look things up, all right? Let's say I do Eichenbaum 2008. Savage. Let's see here. So this is an example. Let's see here. So this is exactly what I was talking about. So let's see if we break this down. So here's what they did with the rodents. You want to know, how do you test a rat's cognition? Right? And so remember we talked last class, animals like to smell stuff a lot? Right? Give the example, Juno likes to go outside and check her pea mail around the neighborhood. Right? Leave messages for her friends. See if anyone left a message for her, etc. Right? So rats are really good with their odor memory. They can remember stuff really good. And in fact, way more with odor memory than they are for visual memory. So us, we're very visual creatures. We give memory tests using vision a lot. Memory for words or places and things. So in this case, you give them smells. So something might smell like thyme or basil or lemon or cumin, right? You guys know those smells? Okay. Would you know if you smelled it before? Yeah, right? If I came in here and I spilled a big old jar of Mr. Clean lemon cleanser, y'all would know it. And if I asked you, if you remember that smell later on today, you'd probably say, yeah, right? Okay, so we're doing that with the rats, and they notice that there's different kind of beddings inside each of these cups, right? Or what have you, actually different. So you have wood chips, beads, sand, screen, etc. And later on, after they learn that information, you wait about 30 minutes, give them something else to do, kind of distract them, and then later on you come back. And you're going to test them with some of the old things, so thyme, cumin, but you're also going to add some new smells, like cocoa or coriander, or whatever that is.
Cilantro.
Huh?
And cilantro.
Cilantro? Why don't they just say cilantro? Okay. It's like, you know, Erica, you know, so we're planning our wedding thing for July, and she's going through all the, you know, how people plan weddings, there's like stuff to go through, right? So she's looking at like colors for things, and there's things that people have invented called like mauve and salmon, and I'm like, why don't they just call it pink?
There's lots of pinks.
It's just pink.
No, there's not.
So yeah, right? So okay, so we got coriander or whatever that was.
Cilantro.
Cilantro, yeah. Which is delicious. actually really like cilantro. But the point is, right, that's a new thing and a new thing, right? And so the rat looks at each of those individually. Now what they learn in their cage, right, is they learn that if it's an old item, that there is a reward that they like near and dear to their heart, a Fruit Loop, right? So if you're a rat and you're bored out of your mind hanging out in a cage all day, a Fruit Loop's kind of a big deal in your world, right? So, especially if all you get is normally like healthy, boring food, right? So Fruit Loops a huge deal. It's loaded in sugar. It's just like a diabetes delivery system just waiting to happen, and they love it, right? So it's delicious, and they can't wait to get this Fruit Loop. And so they learn that there's a Fruit Loop in a cup like this, in the bottom. They have to dig through that bedding to get that Fruit Loop, if it's an old item. But they also learn that if it's a new item, they can still get a Fruit Loop, but it's going to be at the back of the cage. And then they go to the back of the cage and they press a little lever, a little Fruit Loop comes out, and they go, oh, great, thanks. And then you start it up again. You test different smells, right? So what you can do, remember we talked about shifting criterion, right? Your sort of threshold. So we're changing that relative balance of work and reward. How many of you guys would be ready to, I don't know, think of all sorts of stuff. Have a foot race right now across campus from one building to another if I offered $1 to the winner? Why not?
I don't like trying.
Yeah, but it's $1.
It's just $1. You can keep your $1.
Okay, well, I was about to offer $1,000.
Okay, that's different.
Oh, but you just told me you don't like to run.
Like $1,000.
Oh, okay, so if we change the relative amount of reward, then it'll change your behavior, right? Well, what if we kept it at $1, and instead we said that you don't have to run to the other building across campus, you just have to run to across this room. Still win. What is $10? Would you run from this side of that wall, this wall to this wall for $10? Okay. Yeah. So see, so now we're not just changing reward; we're changing work, right? So there's a difference between running between this wall and this wall to running across campus. There's a difference between $1, $10, and $1,000. So that's what they're doing to the rats. They're saying you can change the workload, how deep these cups are, so that it's not much work to dig through there. The rat just kind of goes, right? And you guys know lazy animals? If you don't, I'll introduce you to Juno again on next Monday. Real lazy. You know, like she's gotten to the point where we'll try to we'll give her some treats sometimes, and she'll just be like, bring it to me. I'm like, all right, princess. And then we bring it to her, right? Because we're enablers of that spoiledness at this point. But the idea is they can scoop through this real easy. Now, you can do that easily. Now, you could change the relative rewards for the old items. So, sure, you might not have to do much work, but you're not going to get a whole lot of reward. On the same token, look what we can do. We can change how much they dig, a little deeper. They really have to dig a lot. So if they're all the way digging to here, right, for just a tiny little baby bit of Fruit Loop, that must mean they're highly confident. That recollection, cognition, right? Especially if they know I'm only going to get a little bit of Fruit Loop, a quarter, right? 25 cents of the Fruit Loop. And they have to dig all the way down there. They got to run a whole lap around campus just to get a 25-cent piece. Or you can do nothing, not run around campus at all, stay here and get 3 whole Fruit Loops, which is 12 times as much as this. So you can see that relative mix. And that's how they very powerfully leveraged that experiment to understand the role, say, the hippocampus in those cognitive-related processes. Pretty ingenious, huh? You guys got to convince Dr. Amadeo to do that task in his rat lab here on campus. I think he has mice, actually, but yeah. I've been trying to get him to do that for a while, so maybe you guys can convince him. We'll do some memory studies. I just think it's a really cool way to get at it. So we have that. Good question. Okay, so let's take a look at some of the other memory-related stuff. So we talked a little bit last class about patient HM, and hey, so he had epilepsy, and one of the ways that they tried to treat it was with pharmacological treatments, and it didn't really take, and so they did some surgery, and what they did was they removed his bilateral medial temporal lobe. So hopefully this is review for you guys. This is a little snapshot of him during that time. And so what happened was what? Who remembers?
He couldn't form new memories.
Okay, so he couldn't form new memories, and particularly he lost the ability to form new memories that were consciously accessible. Right? So this is a little cartoon that kind of shows the area of the brain that was particularly affected, this medial area of the temporal lobe that focused mostly on the hippocampus but also involved parts of the amygdala and other areas of the entorhinal cortex. And so we've got this hippocampal shaped structure. It kind of looks like that seahorse, which is where the word hippocampus comes from. It's Greek for seahorse. And this is a ventral view of the brain, so you're looking at the slices this way through that, so that's a coronal view. And we see a normal person's hippocampus would be kind of nice and full in here, whereas there's all these deep sort of black holes where the hippocampus would be in patient HM. And you can also look in sort of this more posterior slice of the brain as well. You've got these areas, parahippocampus, hippocampus, and we can see that they're pretty shriveled up over there. So we have that idea of where the damage was, and he couldn't form these new long-term memories. And it gave this idea that we talked about last class, retrograde amnesia versus anterograde amnesia. And so particularly, he had pretty decent memory for short-term information, but if you asked him to remember that after a period of encoding, it tended to fall apart. So let's see here. We have that severe anterograde amnesia, and we can start to see some of the ways that that became evident. So this shows some of the learning that he still had the ability to do. All right. So how many of you guys know about the mirror tracing task? Okay. So Sean, what was he doing here on the mirror tracing task?
So they give him an image like they're showing a star, and he can't directly see it, but he can see its hand in the mirror, and he has to learn how to trace the star.
Yeah. So one of the key parts, good, exactly right, is this easy or hard?
Hard.
Yeah. Like imagine having to. I mean, it's just weird, right? So what do we? Who knows what we're looking at here on the y-axis?
How often he messed up?
How often he messed up. So we're looking at errors per trial. What's the x-axis?
Number of trials.
Number of trials. So basically, how many times did it take before he messed up less? Right? So in this case, yeah, about 3 or 5, and then he started getting better after about 6 or 7 or so. Right? Okay, so this is day 1. What happens in day 2?
He didn't remember the task, but he was able to do it
Yes.
still.
All right, so you said he wasn't able to do it, and then I was going to ask, but could he remember it? But you already answered. You're like, yes, he could not remember it. So his performance got a whole lot better. So he got much faster at doing this task. Right? He's about where he left off, and it gets even better in the 3rd day. But importantly, you said, oh, cool. So would you study this, or you remember doing this before? He's like, no, I've never seen this in my life. And so it showed us through behavior, just like the rats, right, that there's ways of memory that we can see evident and that we can ask for, and people can show us, besides verbally telling. Particularly, he couldn't verbally remember any of this. That is, he couldn't say it in an explicit way, but he could show that he did it, and still not be consciously aware that this learning had occurred. So it gave us this really classical distinction in the field by the difference between conscious and non-conscious memory, and particularly giving us that distinction as played out upon different anatomical structures in the brain. That is, saying that the hippocampus was evidently necessary for these conscious memories, but because he didn't have a hippocampus, it was evidently not necessary for these non-conscious memories. Seems pretty compelling, right? So if you were drinking haterade on this study, what would you say to criticize those kind of interpretations that we just talked about that we made from this finding? What's a limitation of it? Yeah.
One subject?
Yeah, it's only one subject. People are weird, right? How many of you guys have like a friend or a family member who's just kind of weird? Yeah. If you don't know that person, it might be you, right? Like that's like there's always one. Right? Sometimes there's odd stuff in nature, right? And there's weird patterns, and we really like to see replication. We like to see bigger group effects, even though sometimes patient studies can be very powerful in demonstrating something, even only one time. So, for example, how many flying pigs does there need to be for you to know that pigs can fly? One, though you kind of like to see it again, right? Just make sure you're not the crazy one, right? But you guys are right? So I mean, you kind of just need to see it once and just be like, all right, so I guess they don't fly, or they do. Yes, instead.
Related to this, so in this case, the hippocampus is not needed to do non-conscious tasks.
Yes.
So then would that mean that we don't need the hippocampus to use procedural memory? Is that
Yes,
what
that's
the-
exactly what they.
Oh.
That's exactly what they take it to mean. And we'll that goes kind of right to where we're going next. And so we've got this idea that we talked about, this difference between conscious and non-conscious memory, right? Explicit versus implicit memory. Or the other way you could call it is declarative versus non-declarative. That is, memory you can declare explicitly versus memory that you cannot declare, but yet which might be evident through behavior. And so that's kind of the procedural memory that Lisette was asking about a moment ago, right? So that is memory that you can show by performance as opposed to consciously recollecting it. Yes, sir.
So, I asked this in peer lab, couldn't get an answer, unfortunately.
Uh-huh.
So anterior grade amnesia is what happened with HM
Uh-huh.
in the hippocampus. He can make new memories. So with retrograde, what part of the brain would that affect?
So you're asking about what part of the brain is affected by retrograde amnesia, and I think the better way to think about it is kind of to reverse the relationship of some of those terms, which is to say, where in the brain would you need to have damage in order to have retrograde amnesia? So retrograde amnesia by itself doesn't affect any area of the brain. It's the areas of the brain that end up giving you retrograde amnesia, for example. Does that make sense?
No.
Okay.
Can you repeat it?
I said, retrograde amnesia does not give you any effects on the brain. It is the effects on the brain damage that give you retrograde amnesia. You again?
Yeah, no, I got it.
Okay. Again.
Can you give an example
Of that or of what Roman was asking?
of that?
I can try. It's a good question. So, if it's sort of an issue of sort of causation, right? So if you have retrograde amnesia, it can't cause you to have brain damage.
Oh, okay, thank you.
But brain damage can cause you to have
Retrograde amnesia.
retrograde amnesia, right? So it's kind of like, yes?
So I did read in the book that there are a few things that can influence retrograde amnesia, and it's like damage to the basal frontal cortex and just the cortex in general. Yeah?
Yeah. Yeah. So, so the idea that, let's see here. I can put this up a little bit. so let's say that we have this model here. Okay, so you guys are presumably familiar with this, and we have, so we're just talking about long-term memory, by the way, right? Because there's different kinds of memory. So you can start here with, like, say, sensory memory. Right, sensory memory is going to go to short-term memory. And short-term memory gets encoded into long-term memory through a process that we're going to call consolidation. Okay? And long-term memory can cycle back to here, you're going to come back, you can have back and forth. But the idea is, what we're talking about here is this stuff. Long-term memory, and we are not talking about sensory or short-term right now. Okay? So, we focus just on this part. When you're learning these things that we talk about, we can start to see that people characterize the declarative and non-declarative side, and to the question, okay? The idea, let's say you have your brain, and you got your hippocampus in here, okay? So your hippocampus might be an area that could be involved in, say, we talked about anterograde amnesia. For the retrograde amnesia, the idea is that you can't remember stuff that happened much earlier in time prior to the damage. And so you might have, areas of the, say, frontal or parietal cortex could be one candidate area that could have damage, because the idea is that the information comes in, right, from sensory to short-term to long-term, and that long-term memory is going to be basically encoded by the hippocampus, and then what? Well, the idea is it thinks about it, maybe you go to sleep, maybe you get some rest, it gets kind of consolidated into this stronger memory that after a period of time ends up not being activated in the hippocampus, but instead that memory that gets consolidated into this robust long-term memory is actually kind of supported by activity then in the cortex instead. Usually the type of cortex that was involved in processing that sensory modality. That is, if you were studying music, it might be in the auditory cortex. If you were studying words, it might be in the temporal lobe cortex. If you're studying visual pictures, you might have occipital cortex or parietal cortex activation. Yes?
Sorry, so what you're saying is that the hippocampus doesn't store memory?
It can be involved in storing memory. So this is a good question. It's kind of a deeper question. And it can be confusing at first, because a lot of textbooks and teachers oversimplify it. But, there's a danger there. When you oversimplify something too much, it's not true. Right? And so the hippocampus is involved in storing memories. The information comes in and it gets bound up and packaged by the hippocampus. And then it is involved in taking that packaged up information and sort of exporting it out to the cortex, where it can be stored, so to speak. And the hippocampus is kind of like a librarian, right? You bring the library, you bring books to check back into the library. You guys remember libraries? Yeah, well, we're in like the digital age now, right? Like, I didn't know if people still go to those things. Like, I used to go when I was a kid, but I mean, I don't know. I read and write now 1,000 times more than I do when I was a kid, and I never go to the library. So, just want to make sure that they're still out there. But, so a long time ago, they used to have these things called libraries, and they had these things called books. And you could take the books, right, and you could rent them, and like you could turn the pages and everything. It wasn't a Kindle or, yeah, it was crazy. Kindles are great. They're fun. I'm just starting to learn about them, and it's like, you know, like, oh man, they seem pretty cool.
I like reading books.
Yeah.
It's a thing.
Yeah, it is actually easier. I like it easier on the eyes than the digital screens. So, I agree with it. Anyways, jokes about books and libraries notwithstanding, the idea is that you take a book, you bring it back to the librarian. What is he or she do with it? Well, he's got to organize and put it back in the shelf somewhere, right? Okay, so now you go, so that's kind of like the hippocampus encoding information in. It encodes that book, it brings it in, but it has to then store that book in an organized way, right? So it has to come up with a number, and it has to put that number in ranking in an organizational system. Then, sometimes later, you need to remember that information. You come back to the library to go rent another book that somebody else had brought back, right? One second. The librarian would go and use its accounting system of wherever that organized Dewey Decimal number is, go find that book wherever it is on the shelf, because it knows where to find it because it had sequenced it, pull it back from the shelf, and give it to you as an output of its memory. So the librarian is like the hippocampus, and the bookshelves are like the cortex. Myra had a question, and then I'll get you to say.
So, I think I just like the way that you just explained right now, because I don't know if I want to even say this anymore, but, so the hippocampus is similar, not exactly the same, obviously, as like the thalamus, where it redirects the information where it's supposed to go. It'll absorb it first, and be like, okay, here, this is where, you know, for short term, and then it has to go to, if it's like advanced move, it's gonna go to like, you know, your primary motor or something like that. Similar to that, right? So
It can be, yeah.
it'll take it and then put it to where it's supposed to be, so similar to the thalamus where it receives information and then sends it to where it's supposed to go.
Yeah,
Similar?
yeah. And it depends on the timing, right? Because it's like, it will be kind of like in the hippocampus, so to speak, lack of a better term, for a little bit of a period of time, until it gets really, really strongly bound up, and then it's kind of becomes cortical. So when you would have, that's why you would see retrograde amnesia deficits if there's damage to the cortex. Is because you're going to have deficits in remembering the stuff that was stored in that area of the cortex. So does that kind of help with your earlier question? Okay. Yes?
So how does that work with patient HM? He has the information stored, he gets progressively better with the non-declarative memory,
Mm-hmm.
but he can't access it, but he knows it when he does the task.
Yeah, so the idea is that he's not, that the hippocampus again is doing its job, exporting that information to the cortical areas, and he doesn't have the ability to encode that information in a way that he can retrieve it in a conscious way. But it evidently has systems in place to bypass that and encode particularly motor and other sorts of procedural memories into the other subcortical areas that might not rely on the hippocampus for that processing. This is the big storyline for a long time. Yes?
So I was trying to find a way to phrase my question. So tying it back to your analogy of the librarian and grabbing the book off of the shelf, is that process considered like the retrieval path? And then
Yeah.
is
The
that,
second half of it is, yeah.
okay, so it's part of it. Okay.
Okay, so we'll come back to this idea because there's some useful figures for that along the way. Let's see here. Okay, so ways that we've tested this include things called like the delayed non-matched sample task, and let's see if I can put this down for you. So in a delayed non-matched sample task, this is a way that they've been able to study these kinds of memories and look at the effects of damage to the hippocampus in other species like monkeys, for example. So monkeys really like stuff like treats, just like Juno and you and I, they like M&Ms. Actually, Juno doesn't like M&Ms. She's not allowed to have chocolate. Dogs don't do chocolate. So, little PSA there. If you have dogs, don't feed them chocolate. It's not good. Erica, I get in trouble with Erica if she found that what I said to Juno chocolate. So, anyways, so delayed non-matched sample. It's going to kind of look like this. You have a monkey. It's hanging out, right? It's probably a bad monkey because it's in jail. And no, they're bad, man. You should. You guys ever see if you can go to certain places. I think there's like resorts in India where the monkeys will steal your cocktail drinks. Yeah, they end up in monkey jail. And so there's two things here. You've got basically an object on their left and an object on their right. Okay? So you've got the key and this little bubble, right? Now, underneath these can be a tree. Okay, so it learns this. So you have a learning phase, and then you wait a little bit, a delay, then you give it a test. Okay, so you show it these stuff again. And so you're testing. Remember we talked about the rats a little while ago? How do they show us their memory? Same idea here. How does the monkey show us its memory? Because if you ask it what it remembers, what's it going to tell you? Say, "Ee-ow!" That's how the monkeys, that's the only thing they can say to you, right? Okay, so you're not going to get very far asking it, you know, how do you feel about this? It's going to take its poop and it's going to throw it at you. Okay, and they do. So what you're going to do is you're going to ask it to show you what it can remember. So you're going to say, I will give you a treat under the thing that was not here before. So it's a non-match. So you give it a sample, then the treat is under the thing that does not match to the sample after there is a delay. Which is why it's called a delayed non-match to sample. So it learns that there's these rules of that, and so when you test it, if it grabs the key, because it thinks that's where the M&M is, it shows you that it did not remember either the rule or the location, but one of the two, there was a memory failure. You guys follow the logic? Okay, so that's called the delayed non-matched sample, and you can see data like that. When you do that, you can score their memory on the y-axis. So we're looking at here. How good is their memory? In the control group, healthy normal monkeys, memory's pretty good. Let's say that you can have different types of lesions of these medial temporal lobe areas. Now we've talked a lot about the hippocampus, but let's talk about some other areas that hang out with the hippocampus. Okay, we're going to call areas like the entorhinal cortex, the perirhinal cortex, the perihippocampal cortex. There are these areas shown in these different colors. Okay, so you've got this sort of hippocampus here, perihippocampal, perirhinal, and entorhinal. So it's just a cartoon showing kind of generically where these different areas are, but they all mush together right next to each other to form this network of subregions in the medial temporal lobe. Because it turns out in the time since patient HM was discovered in 1957, we've learned that there's a lot more special areas in the brain than just the hippocampus. Right around it, there's these other areas too, and they actually matter a lot. We're going to see that memory breaks down differentially across these different areas. So, if you look at how is the memory, right, in just the hippocampus, when the hippocampus is chopped out, turns out they can do delayed non-matched sample pretty darn freaking good, right? Turns out if you want to get memory to be really bad in that monkey, you've got to cut out these extra areas of the medial temporal lobe. So you've got to take the hippocampus cut out, but plus you've got to add, the hippocampus has to add some other areas that are also chopped out. So you have these little plus signs for added areas beyond the hippocampus which are damaged. You guys follow the logic? So this is work that was done in large part, I think, down here in Southern California at UC San Diego, mostly in the 80s. I don't think they labeled this in a way that was very intuitive. I never really understood. I thought it meant the opposite, which is why I tried to explain it so it makes sense. Yeah.
So did they have further studies where it took out only the entorhinal or only
Entorhinal.
or the other one as opposed to just adding it with taking out the hippocampus?
Not in this particular study, but they have since. Yeah, it's a really good question. But in this particular study, again, this was basically in the 80s with monkeys, and they just kind of did it in this progressive fashion. These were the results that you had. Now, Roman's been working in my lab for most of this year. Constance, you joined this semester. Lindsay's been with us for about 3 years now. And I imagine that many of you guys are probably have worked in other labs for somewhat different numbers of lengths of time, years maybe, right? If you got done with your work at the end of 3 years, Lindsay, and you just did all this work, and then someone asked that really, really good question. And they're like, well, did you do it this other way? You'd be like, no. Right? You're like, this is what I did, and this is what we got. So let's work with this first, and we'll do the next study later. That's exactly what they did. So these became prominent studies early on because it takes a long time to do these right. So they did this, these became prominent figures, they probably just became legacy figures in textbooks and things like that.
I was just curious if like the other parts of the brain taken out on their own would have affected it, or if just the overall had the negative effect.
That is a very good point. Does anyone have any idea of what a possible answer to that might be?
Look at the research.
Yes, Rosie?
Well, I was reading, and it says that parahippocampal cortex plays a role don't know if it plays like a significant role, but in processing contextual aspects of memory. So, I don't really know what the surrounding text is, just what I wrote, so I don't know, does that indicate that it plays a role in specific types of memory?
So what I was doing, I was fishing to see who remembers some figures and stuff that the text talked about. So they're coming right up upon what you were saying here. So, let's see here. So this is actually data of exactly what you were talking about. You said, has anybody chopped out just the parietal cortex? Yes is the answer, except yes, comma, rarely is the answer. Because it is really freaking hard. Particularly, what we're looking at here is very special data because of how rare it is. It's from a human, it's not from an animal. And this is a reconstructed medial temporal lobe of a human who's had brain surgery. Now, medial temporal lobes are bilateral, right? There's one on the left and there's one on the right. See, this is the left and this is the right. So we're looking from the outside in here. We're looking from the inside out there. Okay, now this is patient NB that was operated on in Canada, actually by a friend of mine, Ben Bowles, did the research. He made this figure, actually. I reconstructed her MRI slice by slice by slice with different colors. It was his dissertation work. And so this single patient had epilepsy, not unlike patient HM, that was localized this time, not to the hippocampus, but to the parietal cortex instead. And so the parietal cortex is basically this sort of yellow area here, okay? So the entorhinal cortex is in green, and you can think about it in this way. So one way to think about this medial temporal lobe system is kind of like a pyramid. At the top of the pyramid is the hippocampus. Okay, now the hippocampus has input that's given into it by the perirhinal cortex. It also has input giving into it by the parahippocampal cortex. Okay? Now the parahippocampal cortex and the perirhinal cortex, they talk together and they send their information into the hippocampus, but it cannot go to the hippocampus directly. Who knows the name of the structure that's necessary to get into the hippocampus? Not quite the mammillary bodies. It's called the entorhinal cortex. Okay, so this is your entorhinal cortex. Entorhinal cortex, and the entorhinal cortex is going to be the proverbial pipeline of anatomical connectivity into the hippocampus. And so I draw this diagram to help understand what we're looking at in color here. Okay? So hippocampus is in blue, the amygdala is in red, which you're not really talking about right now for memory, but this is your parahippocampus here. The green is the entorhinal cortex, and the yellow is the perirhinal cortex. Okay? And so we can see that there's lots of green. The perirhinal or the entorhinal cortex is intact in this patient. But the patient had particular deficits in just the perirhinal cortex, just like you asked. Now this was done, I think, published in 2008 or 2009, maybe. This is, 40 some odd years after patient HM. Took a long time to find a very careful lesion of this type. But they found it when they tested it. They found that the type of memory deficit that this amnesia patient had was a very, very selective type of amnesia. It was just amnesia for this type of memory we call familiarity, not for recollection. Okay, so we back it up here on the slide, and we see that what we have introduced was this more modern view of declarative memory. So it wasn't just declarative and non-declarative memory, but it turns out that within declarative memory, there's different kinds of episodic memory called recollection and familiarity. Each of these cognitive processes rely on different neural brain structures, such that recollection seems to rely on the function of the hippocampus, but familiarity relies upon the pararhinal cortex. Evidence for which we just saw in that patient NB. Okay. All right. So there's our patient NB again. I'm going to skip over Korsakoff's for the moment now. And look at this different sort of other subdistinction that we can find in declarative memory too. Now we'll bring this back in a moment or two with a figure that shows a nice little diagram of these different kinds of memories that you guys are probably familiar with this declarative memory model. Okay? But the other big distinction is this distinction between semantic memory and episodic. So semantic memory is your memory for just basic facts. So what is this? Or soda. But either way, yeah. That's semantic information, right? It's just a general knowledge and world facts. Episodic memory is your memory for the episode of when I bought it, or when you dumped it on your head, or the first time you drank it, or when it got you sick. That's an episode of your past. Now, the episodic memory relies upon the hippocampus. That's basically what we've been talking about. Semantic memory seems to be reliant on more cortical areas, particularly the frontal cortex, as well as the temporal lobe cortex as well. So yeah, we have this sort of classical distinction between semantic and episodic memory. And patient HM had damage to the hippocampus, but he did not have problems with semantic memory. So it shows they're kind of different, for example. Now, let's see here. So we look now on the other side to the non-conscious memory. And so we can talk about some different types that exist. So we already talked about some basic skill learning, such as that star tracing task. You can also have different kinds of priming. So you can prime someone with concepts. You can prime some people with semantic information. You can prime people by just repetition priming. That is, just having something be repeated might make you more likely to remember it, even if you can't say you ever saw it before. So one way we would know that is if we measured your memory for something that you don't remember seeing before, but you have a faster reaction time to it than new things. Right? So your behavior is showing that you learned something. Lindsay's done a big study here on semantic priming. So you can have words that are related to each other, like doctor and nurse. And you can prime people so that they say hospital or something like that. You also have associative learning. This is kind of tricky in that associative memory seems to be in declarative associative memory seems to rely upon the hippocampus. Non-declarative associative memory seems to not rely upon the hippocampus. That is, certain forms of habituation, cell level learning that we'll look at with some follow-up slides later on. So classical conditioning, for example, You guys know Pavlov's dogs? You know, remember Pavlov, the guy with the big beard? You guys know why his beard was so soft? It was classically conditioned.
Oh,
Yeah, I went there. Yeah, could have had a whole class with no jokes, but at least they got the lame ones. Yeah, Rosie.
I totally missed what you said. You said it's the non-declarative or the declarative?
Oh, I thought you wanted me to repeat the Pavlov joke.
Oh, no. You can, but.
No, I don't think they want me to. I'm sorry. Go ahead.
Is it the non-declarative or the declarative that relies on the associative?
Declarative. So things like associative memory, like if I'm asking you to remember 2 pictures or 2 words.
Okay.
But, you know, again, so some of this stuff is going to get kind of confusing, and I admit that at times because the field is limited in our language. So we call things associative memory at different, so that's a kind of associative memory that we even study in our lab. But you can also look at Pavlov that we just talked about, right? And he also has classical conditioning, which is learning associations. And that's not always relying on the hippocampus either. So it's just because we use it, it's just because the language we use kind of makes it a little confusing at times. So, sorry for that. Fair warning, it's not my field. I didn't invent it, I just work in it. So, yes.
just based off what you said right now, so associative learning is both for declarative and non-declarative? Because you said.
It can be.
Oh, okay,
It's
okay.
listed under here as
That's right.
non-declarative.
Right.
I try to avoid test questions like that, because I look at it and I'm like, this is too dumb for me to ask my class, because I think it's a dumb question. And I know the field well enough to where I'm like, this question makes no sense. And so I try to hold those back. You guys have already caught me a few times where I made some mistakes and I let a few slip in there. But we fix those. So, no harm, no foul. But yeah, I'm just trying to clarify some extra information beyond the textbook that, you know, I can give you from being directly working on the front lines in the field. You know, textbooks are just starting points, right? I mean, they're not the be-all and end-all of anything. They're usually wrong in some things, like we just kind of picked out a few little details of. And we hope that they are, because we hope that we're discovering new things. It takes a long time to put something in a book, because you gotta be sure of it. So by the time you're sure of it, you might be figuring something new out. You know? Anyway, so yeah. Speaking of test questions, you guys have any good ideas for test questions I can ask next time? All right, you'll just get my questions again. Oh, okay.
Not actually a test question, I actually just have a question about this. I was reading about patient MB, so it's totally unrelated to that, I apologize. But it says that part of the amygdala was also lesioned out, so does that mean that he also had like problems with like emotional attachment learning?
Good question. So parts of the amygdala were also lesioned in patient NB. Happened to be a lady. I don't know that they tested a lot of emotional memory on her. So I don't know that we as a field know the answer to that. It's a good hypothesis that it's likely she might have had some damage to that type of memory. I'm not sure that we tested that as a field, and if they did, I don't remember what the result was. I didn't come across that finding. So, good thinking though. Yes, Rosie.
Possible test types.
Okay,
You could
all right.
ask us what types of, or like, if you do like short answer again, you could do like, what's the difference between like declarative and non-declarative, and like just have like for the various different types, sort of the different, I don't know what those are called, like subfields of it.
All right, that's a good idea. So what's the difference between declarative and non-declarative and different subtypes? That's good. Maybe draw out the memory model that we're gonna see that's coming up that I'm sure you guys read ahead of time. That could work.
Draw it out on the computer?
I don't know. I haven't thought that far yet.
I mean,
Maybe. I don't know. Maybe they let you do that. There's, you know, Blackboard's got some powerful stuff. You probably just take your little cursor or your touch screen and, I don't know. I doubt it. You're right. It's probably funky.
subjective grade bond.
Yeah, so the reason I ask is because I've been trying to think of ways to be creative in helping your learning and studying, as well as your test performances.
Why don't you ask us the day before the exam
About what?
about what questions?
Well, because if I ask you the day before the exam, you're not gonna have time to get them to me, and I'm not gonna have time to put them on the exam.
That's when you
So-
should take the exam.
Plus, it shouldn't matter because you should be studying just as much now as you are for the day before the exam. Theoretically, I know that that doesn't always work out that way.
I know, but just so we could go over like all the topics.
Okay, all right, that's a good point. So how about,
Labeling diagrams?
I'm
Did
sorry?
we say that?
sorry,
Did we say labeling diagrams?
maybe? Could. So what I'm trying to get at is this idea that I've been thinking of, see if you guys like it, that maybe we can do some sort of an assignment that would count as a quiz or something where every person brings in 10 test questions that you write. They have to have answers that you have to provide that are correct. Yeah, you see what I'm saying?
Do you want page numbers too?
Page numbers could be helpful. You might want to do that in case someone wants to question your question.
So that's a
Just gonna
maybe.
put all of my answers in.
No, put a question, put page number by it, yeah, sure. Yes,
If we're doing this, are we all getting provided with like the classes questions so we can study off them? Or is it just
yes,
going to
but they have to be correct.
you?
So actually did this a couple years ago, actually during Lindsay's first class year. We did this with your quizzes. You guys would submit your quiz questions in biopsych. You probably don't remember that, but you did. Trust me. She did. So we would have quizzes where the students would come up with the questions, and then I would add my own, and it was a mix, and everyone would have access to everyone's questions. So that you all had to study each other's questions.
So we get access to yours too?
You want access to my test questions?
Yeah.
What's your
Are you
test
sure?
No, you didn't make them work.
No, I promise I won't make them harder. But now you're gonna get what you asked for. So this is what we'll do.
Wait, just to understand the terms, if you provide the questions, would that mean that you then change the questions?
No, it doesn't.
Okay.
So we just have to learn everyone's questions in yours?
That's all.
So we're
So it's
writing
like a
down.
study
You don't
guide.
need the full test paper.
I feel like there's a,
I feel like,
there's
yeah,
a
this is too
catch
much.
to this. I
You're
know.
gonna give us like 300 questions.
You guys are thinking good. I like it. So should have listened to Lindsay. But no, so seriously, guys. So what I can do, I will give you every single question from the test bank. How about that?
How many are we talking?
So they're possible
How
questions.
many?
Yeah, I'm not gonna give you the test ahead of time. I'm dumb. I'm
No,
not
no,
a
right.
cheater.
Just to understand, so they're possible questions, and all of the ones that you provide may or may not be on the test.
Yes,
There's nothing more.
let me get to it.
Okay. I'm trying to
Just relax for a second. We'll get there. So the idea is this. I will give you every single possible question for all the chapters on the exam. Okay? There's a lot. And there's no way I could possibly put all of them on the exam. Unless you guys are just truly masochistic. Like there's hundreds. So it's not gonna work, right? Like I can't test you on those. Even if I want to, the school would like make you leave class or something after a period of time. So so that's the point, right? Is I can't put all of them on there. You don't know which ones I'm gonna pick. Now I don't know which ones I'm going to pick until I pick them. But you'll have an idea. And it also gives you something to study from. This isn't actually a new idea. This is actually how they test every airplane pilot you've ever had fly your airplane. So the FAA publishes all of its test questions on its written examinations. And as a plucky young student pilot, I was all excited about that, as most young student pilots were. Like, oh yeah, sweet. I got this in the bag, this will be easy. Turns out the joke was on us. Because they only test you, I think, on the FAA on like 50 questions or 60 questions, but there's like 400 questions in the test bank. So you don't know which ones it's going to be, which means you got to go learn 400 questions. Which means even if you learn what the answers are to the questions, guess what? Joke's on you, you just learn. Right? Which is exactly what the FAA wants, you to learn the information. And there's no way to know it, there's no way to get a good score statistically by just memorizing question to answer. Because the options, A, B, C, D, change for every person. Yeah. The information's the same, but they get shuffled in there. So you can't just learn it an association like that, associative learning, and that you actually have to know it. And so if all you do is rote memorize that information, say for the FAA, and you go take your test, you perform at about chance level. So, you know, that's a fail. So you actually have to know it, but it's a useful guide. And so if you want, I'll give you that's how confident I am with the test, is I'll just give them to you. You like that? Okay. Yeah, I mean, you know, it's not gonna make it easier, I promise you that.
No, but it'll give me an idea of what I'm looking at.
Yes, but so the questions are written by the same people who wrote the chapters. So you already know what you'll be doing by just looking at the chapters, but it'll be a useful guide. And I'll do that.
It'll just help us get rid of the excess jargon that they throw in there that's not. No, some of it over-explains stuff that we already know. Like in chapter 1, if we were talking about look, this is just an example, but not everything's jargon, but if it over-explains something that wouldn't necessarily be tested on, or that we as people might not use for our field in general, then it would be nicer to find, like.
Yeah. Although, you never know what you're going to use in the field. So when I took all my classes in grad school, it was all like cell and molecular biology, and I was so happy to get done with it because I'm like, I'm never going to use this. I'm going to do cognitive stuff with EEG at the scalp entirely, so I'm never type to have to do this again. And when I ended up doing that NASA mission, we did all the exact same stuff they taught me in the UC Davis training program that I swore I would never use again. Because I'm like, I'm not going to do molecular biology. I'm not going to do DNA sequencing. And then NASA's like, hey, can you go do some DNA sequencing? And I was like, okay. And it ended up being really useful that I had that training. So
But I mean, like more so some of the studies that they put in there to kind of give us examples.
Yes, but here's the thing.
But not like
We're
that
also
we wouldn't use, but that wouldn't
So here's the
really
thing.
help us learn. You know?
I'll give you all these questions, and you're right, it will be helpful. I wouldn't be doing it if I didn't think it would be helpful for you. But it will also include some questions that you guys write. The test will include some questions that you guys provide with answers and questions too. Now, I'll review them and all that, so yeah, question on that.
So essentially, just so we can get an understanding, you still want us to do the 10 questions. Those would be, as an assignment, we can go ahead and do those, also answer them. Those could be a possibility of being on the exam, as well as questions that you already have pre-written. And then you would give those and allow us time to be able to either use them as study tools or whatever. So then have a combination of the two, that's essentially it.
Yes.
Yeah, okay.
Yes, and then yes.
For our questions, are they all gonna be multiple choice? Or do you want them to be all multiple choice?
You can make them any way you want. Be creative. Yes.
So when you said all chapters, is that gonna be for the final because it's cumulative? Or we're talking about exam 3?
I'm just talking about exam 3. I mean,
Okay, because
there's,
you said all chapters, so I was like.
well, for the exam, yes. Yeah, for exam 3 was the context I was referring to.
Oh, okay.
I mean, honestly, I'd probably do the same for the final too, but again, I hate to say the joke's on you in that regard, but it's like, we're gonna have a lot of chapters on it, and there's, so I did this before last exam, I printed, Lindsay asked me for the the questions that could be on the exam, and I printed her every single one, and I don't know, Lindsay, like, there's a lot, right?
There's like 300.
Per chapter.
Yes.
So, I mean, I can do it for the final if you want, but, you know what I mean, like, you can have it. That's why I'm not scared about it, because I can give them to you, and you know what the best thing that happens? You memorize all 300, and you get 100. That's all. Do you know how happy I would be if you guys could memorize all 300 of those questions? That'd be freaking awesome. That'd be like, you'd learn so much. Like, that'd be incredible. That'd be so much better than what we're doing. That's how the FA feels too in that regard. That's why it's like, I don't feel threatened by it. Like, I think it's great. So, I mean, I'll do it for the final potentially too, but let's say we have 10 chapters on it, then that's 1,300 questions. If you got that in the bag, we're good. Yes?
I think if someone got 100% on exam 3 doing all the questions like that, we should have like a pizza party.
Pizza party for,
Neuroscience
Yeah.
a pizza party for what criteria exactly? I could barely hear you in the back. I just heard pizza party.
On the exam, like with like memorizing all 300,
Okay, so you're saying if somebody gets 100 on the exam,
we should have like a pizza party.
Everyone gets a
okay.
Is that what she said?
She said if one person gets 100.
Oh, just
Okay, more
So-
than one person then.
Okay, so yeah, okay, I'm okay with that. I'll do a pizza party if
How about you,
if no, seriously. So you know, she said if one person gets 100, and I was gonna be like, yeah, we just talked about single case studies, and that could just be a weird person, right? So like, we can't do that. But let's say if what's a good round number? 5?
Yeah.
Yes.
25?
No, 5.
5 what?
like 5
People.
People.
If 5 people get 100,
3 people?
3?
That's a lot of people.
We'll meet you halfway for.
Yeah, how many people in the past?
38.
Really?
Yeah, that's what I'm saying. Yeah, not everybody comes, Chance.
Oh, no, they don't.
It shows on the test. It shows on the grades too. No, seriously, if you guys want to know a cool thing that's like a testament for you guys, so I can look on my grades, and I can see basically who turns in, say, quizzes when we're in class. And there's a huge, huge correlation between just physically being present in class, just turning in quiz, and getting better test grades. Just being here is a hell of a start to a good grade. And I mean, I'm sure you're not surprised by that, right? But, I mean, yeah, it shows when people don't come. So, good for you guys for being here. And I hope that it's helping to contextualize what we're reading and clarifying some of this stuff. So, we'll do that. It's easy for me. I'll print the questions. I'll put them on Blackboard. You can have them.
So there's 300 per chapter, right?
But this is just for exam 3, just for now.
Yeah, but I mean, I don't know. I'll probably do it for the final. Trust me, it's not as great of a treat as you think. Like, but you know, it's more of a psychological crutch, which could be helpful too, right? But
I
yeah.
guess to look at it from a different perspective, if you know the material, 300 questions kind of seems like nothing. Even though it's a lot in number, if you know, because in a sense, because you know everything,
Yeah.
you answer these and like they're nothing.
Which is why it doesn't matter if I give them out.
Right.
But the other thing is, right, we're all starting from a point of not knowing the material,
Yeah.
for which 300 is a lot to know. Which is a huge victory if we can go from 0 to knowing 300. I'm like, damn, that's better than what we're doing now. That's great. That's why I think that's why I'm cool with it.
So do the questions include things we don't go over too? Or just-
Absolutely. There's no way I can cover every single thing. You guys already know that. We don't have time to cover every single thing from class. That's been the start since day one, though. That's in every class you've ever taken in your life.
So really you providing us with these questions can only help potentially.
Oh, I mean,
Because they're not due, it's just something that you're posting.
yeah.
What
Yeah.
do you want us to turn in for questions?
I don't know. You guys want to do a quiz next Monday?
Sure.
And just type it in?
I'll be here, sorry.
So, good question. Let's turn those questions in Monday, because we have the test on Wednesday, right?
What?
Okay, maybe I'm wrong. That's okay. It's okay.
We're in touch chapter, what is it, 19 next?
Douse the torches, pitchforks, put away.
Well, we wouldn't be, we're
Like, gosh, I just made a mistake, guys. Sorry.
No, it's the week after.
Yeah, it's the week after. It's the week after.
It's on
Sorry.
the 15th.
Hands up, don't shoot. Like, goodness gracious. Like,
It's in 2 weeks, the 13th.
okay, yeah, so, yes.
So that it doesn't like impede on class time, maybe like open it for over the weekend so that instead, like, opening, being able to provide questions for the test over the weekend from like Friday to Monday and it's due on Monday?
Yeah, that's what I'm talking about.
Okay.
Yeah, Monday. So, let's have you turn in some test questions on Monday. And let's see here. If you're not here on Monday, you can email them to me. Okay? Does that work, Myra? If you're not here Monday, you can email it to me?
Yeah, so probably irrelevant to everybody else, but I won't be here unfortunately, for the quiz. Is it possible if I could go? Well, I guess we'll talk about it later.
I just said you can email them to me.
No,
Well,
I
that's the
know,
quiz,
but I was
just
talking
the
about the
question.
quiz, and you said we have a quiz.
It will count as your quiz grade.
Okay, sorry.
Just,
I'll stop talking.
no, it's okay. It's okay. Just, everything is gonna be okay. Like, we're not designing this to hurt you, we're designing this to help you. Okay, so turn it in on Monday and it'll count as a quiz grade.
Okay.
Yes, Chance?
The 10 questions we're coming up with are just for chapter 17 or for all chapters?
Anything that is currently, right now, assigned as a chapter that could be on this upcoming exam. I don't remember that number off the top of my head, but just refer to the syllabus for the other chapters that we've assigned to cover. Now, chapter 17 is a massive chapter, physically speaking. I actually told them they should split it into 2 chapters, and they must not have listened to me, but that's okay. There's a lot on there, so we're going to spend a lot of time covering it. But does that sound good? Whatever the syllabus assigns as being the chapters that we're supposed to cover?
Yes.
So you might say half and half or whatever, however you want to pick your question distribution. Now the answers you provide must be accurate. Does that sound fair? Now here's the other thing. It's not as easy as you think to write a good question. So do not wait until Sunday night. It is, because you don't want to write a question that your classmates are going to be very frustrated by and say, this doesn't make sense, or this is problematic, or whatever, because it is tough to write these questions in the right way and not have people want to string you up with torches and pitchforks. So it's, this is why I want to use it as an exercise, is because it really does require us to think when we make these tests and when we make these quizzes. How do we ask the right questions to get at the right information that's relevant? How do you guys feel about that, okay? Cool. So hopefully that'll help.
So we're doing 3 people?
Huh?
3 people?
3 people for what?
For the 100.
I'm gonna say, let's say 5.
Meet you halfway for.
Well, maybe you can motivate your classmates to be better students. 5 is a reasonable number. Okay, 5, and how about this? And if no one gets 100, then you guys owe me pizza.
I'll your beer.
Right? That's the IKEBOB test. It's not just, it's that you gotta dig, but also change the reward. So it's not just that you get the Fruit Loop, but you also have the cost of digging. That's all right. I'm actually lactose intolerant, so whatever pizza you bought for me, I would just share with everybody else.
So then,
That's so sad.
I know. Broke my heart when I found out.
Do you really just like bite the bullet and I want cheese and eat it anyway? Or like, or you
Are there
like-
tabs for it?
There are little enzyme tabs that you can get. So yeah, I probably bit the bullet when I came out of the spaceship for NASA's study when we were locked up for that summer. I think when I came out, I kind of had a hankering for some pizza or something. So yeah, every now and then, you kind of deal with a bit of suffering. I'm sorry.
Maybe I'll just be so happy we got the 500 and like
Well, if you get 5 of them, I'm buying. What are you talking about?
We don't even also eat that.
Oh yeah, well, maybe I'll be so happy. Yeah, maybe. We'll see. So yeah, we'll sort that out. If there's 5 people who can get 100, that'd be awesome. Okay, so
10.
Yes.
Since this is due on Monday, will you give us like an estimate time of when a test banks will be available? Because it's like a lot of questions to study.
Totally different things. Good question.
Okay.
So, what's your name?
Kathy?
Kathy. What Kathy asked, if anybody didn't hear it, was she said, since this is due on Monday, will you give us a test bank to study from? And I said totally different things because what's due on Monday is your questions that you invent. You don't need my test bank for them at all.
Yeah. Will they both be available like the same time, around the same time?
I'll get them available as soon as I can, maybe today or tomorrow. But you don't need that at all for Monday's thing. They're just fundamentally separate.
Yeah.
How
So-
long do you think it'll take you to vet out questions for Monday's day to release tests?
Oh, to put on the test? I don't know.
Like,
To the night
are you
before the test.
releasing the student written questions
Sorry?
to us? Are you releasing like the full list of student written questions
Oh.
to us?
Yeah, eventually. I mean,
Okay. Do you know how long that may take?
Oh.
I tell you what, I will try to, if you can make them digital and turn them in or whatever, I'll try to scan them in and I'll try to post them later on that day or the next day. And, I'll look for ones that stand out to me if they're gonna be problematic, but honestly, you guys would be the ones reviewing it. You guys can email me back and say, nope, don't do this one. Nope, this one's gonna be, nope. This will be a fun game, but I don't know if you guys are realizing how much work you're asking for yourselves. There's a lot of extra stuff, and it's a study, but hopefully maybe really good exercises and really good stuff to be useful for, so, yeah.
On that note, moving on with lecture.
Yeah, we can keep going. Totally.
So we're going back to lecture?
Oh, yeah.
I have a question about, because I don't know if you're gonna talk about it later, about, Korsakoff syndrome?
Korsakoff.
Korsakoff syndrome,
Mhm.
and how it's, due to a deficiency in thiamine. I know it said like if you supplement beer with thiamine, that like the issue might not be as relevant, just because a lot of the cases are alcohol related.
Mhm.
What, and I just didn't Google it because I feel like it's quick Google, what natural like foods would have thiamine in them?
Oh, that's a great question. I have no idea what foods have thiamine. Yeah, I mean, I don't know. I don't really drink that much, so I don't really, Yeah, I don't know the answer to that question, Rosie, but it's a good question. I definitely won't test you on it. Yeah, okay, so anyways, let's talk some more about some of these memory models. Yeah, Roman?
Another question, kind of add on to that, was actually something I want to talk about with why thiamine. It kind of stopped the issue with the memory, but it doesn't essentially help with recovery, I guess. I forgot exactly what's in the text when it's talking about it, but
So the thiamine, why thiamine? I don't know, like, what it, like, nature just has it so that, certain, if you have deficiencies in, I don't know, calcium, it leads to weak bones, and there's reasons for that too, but like, thiamine was associated with this developmental problem that it led to this amnestic deficit. Let me see if it has it up here, actually. Korsakoff syndrome, thiamine. Doesn't really talk about the underlying mechanisms of the thiamine-based deficiency, and I don't personally know them either, because I'm not a medical clinician. I kind of look at the deficits associated with the outcomes. So, yeah. Anyways, what I want to get to is So we talked a little bit about this associative learning, so we'll dial into that a little bit. This here is a operant conditioning box. So this is one example of what can be used to study associative memory, as well as operant conditioning. And many people are familiar with Pavlov's dogs. Maybe we'll have an in-class demonstration with Juno next Monday if she's here. I'm sure she'll be happy to salivate for you if you can bring her some bacon or something. But the idea here, just to remind people, is that you have things called stimuli and other things called responses, right? Now we're going to get more specific on that because there's stimuli that can be conditioned and stimuli that can be unconditioned. Responses that can be conditioned and responses that can be non-conditioned, essentially. And so when you really get into behavioral learning and stuff like that, you can see there's all sorts of granularities and specificities here. But the idea is here you have a stimulus that's unconditioned. That is, let's just start with food in general, right? And so it leads to a response that's essentially the dog salivating, right? So you get a big slobbery old dog, right? And so this would be a response that's not conditioned, right? That's just normal. So by conditioned, what we mean here is basically the consequence of repeated pairings, that something learns as an organism that now they become conditioned to, right? Kind of like Dwight Schrute. You guys like Dwight Schrute? You guys know The Office?
Yeah.
Okay. You guys want to have Dwight Schrute explain this?
Yes.
Okay, let's see if Dwight Schrute can do a good job here. Let's see. So first of all, show of hands, who does not know The Office and Dwight Schrute? And that's okay to admit it, because we need to contextualize this. Okay, that's good. So, okay, so Dwight Schrute is a character in a TV show called The Office. It's a fake reality show, and the idea here is that there are two guys working in an office, and it's like in Scranton, Pennsylvania, which is kind of like a rundown town. It's a paper selling company, which is part of the joke of the TV show. It's this dying company in a dying industry in a dying town, such that, right, we're going to digital, so nobody does paper. So it's like, basically you got these characters that are bored out of their minds and they just mess with and they mess with this guy, who's kind of like a dork. He's kind of like their lovable little brother that they kind of play practical jokes on. So that's what we're looking at. It'll explain classical conditioning, or conditioned stimuli.
What? More Melton? Okay. Melton? Sure. What are you doing? What? I don't know. My mouth tastes so bad all of a sudden.
So, let's see here. Might have another thing here where.
Yeah, the Big Bang Theory for the positive reinforcement, that was so funny.
Oh yeah, I haven't seen that one. Maybe. Let's see here.
I have to reboot again.
So the idea here, let's just watch this again here, right? So you have Dwight Schrute, of course, is our lovable character here, and Jim is just kind of the sarcastic jerk of the office, messing with this guy. So he's wants to run this little psychology experiment, which we see, and you have the stimulus and the response. So the stimulus in one case, is it the Altoid? No. It's the bell of the computer. Where it goes, doo-doo. All right. So Jim can, you can all make that little sound in the computer. So Jim uses that as Pavlov's bell. Right. Now here we're not talking about making the mouth salivate of a dog. In this case, it's making the mouth dry or whatever. So it's actually the response that's going to be conditioned is Dwight's hand coming out. Now, that's the conditioned response, because what is the unconditioned response? Kinda, you could say that. You could also say it's the Altoid, when he offers him the Altoid. So every time there's a bell, right, Jim responds by offering him the Altoid. Right? So you can kind of look at it in a couple different lenses. But the point is that he conditions him so that every time you hear the bell, he knows that his mouth gets dry, which becomes the conditioned response. And then hilarity ensues. So now look at it kind of under that lens if you haven't seen it before.
Do you want an alpha? What do you think? What? One alpha? Okay. Alpha? Sure. What are you doing? What? I don't know. My mouth tastes so bad all of a sudden.
We chose classical conditioning as our psychology experiment topic
So-
because we thought that it,
Anyway, so they got these nice little videos. Apparently somebody made this in class, etc. So anyways, yeah. So that's kind of the basic model that we get to, which kind of leads us to this framework that we've kind of alluded to. So you got this basic sort of declarative memory model, sort of tree. So again, we're in long-term memory here. It can be either conscious or non-conscious, that is explicit or implicit, declarative or non-declarative. Things you can declare you know, like semantic information of world knowledge and facts, or episodic information, like things you remember from an episode of your birthday in the past. Okay? Now you can also have things that you can't declare, but you can demonstrate through behavioral actions in one way or another. Right? Now we talked about these different types of non-conscious memory, involving things like learning skills, being primed, or in this case, classical conditioning as well. So this seems to not rely on the hippocampus, and this seems to rely on the hippocampus. Now the thing is that in more recent times, there's actually a lot of evidence that's challenged this model. So it looks like the hippocampus is actually involved in both kind of memories in different ways. And so that used to be a really good test question of, what of those two kinds of memories does the hippocampus have involvement in? And it used to be a really clear answer. And I have to refuse to be able to use that question because I don't think it's true. I've published findings that say it's not true. So I tend not to ask that question because unless I have answers that show a nuanced understanding of it. So anyways, let's talk about some other things that the hippocampus is known to be involved in with memory, particularly your memory for where you are in space. That is what we'll call place cells. So it turns out that there are some neurons in the hippocampus that will show special preference for firing when you're in special different places in a room. And it's from these different place cell firings that it's thought that animals can kind of reconstruct their sort of three-dimensional GPS map of the world that helps them to spatially navigate. Because as you know from spatial navigation, you can't really do it unless you have memory for those spatial relationships, right? Where you were and how to get around. So if you didn't have that memory, you would be amnestic and you would just be driving around Southern California every day hoping you bumped into your house, right? So we know that spatial memory is really important. The idea is that we think that basically animals and humans, we form this map of the world in our head. So this cognitive map came from this idea of Tolman, who was the researcher. And it's not just place cells, but there's also cells that will fire whenever you cross a particular grid. That is, let's say we're in the room here and you can put a grid down. It's not just this place that it'll fire, but whenever you're in crossing certain intersections of that grids, they'll also fire. And the grid cells seem to be located in the entorhinal cortex. The place cells seem to be located in the hippocampus. And as you can imagine, based on our sort of model, they have anatomical connectivity that talks back and forth to each other. And so those grids layer with the place cells to give a network that can help us figure out our GPS location. And that was such a big finding that actually led to was awarded a Nobel Prize in 2014, which is a really big deal. So the Nobel Prize comes with $1 million award, and it was awarded to 3 people for that discovery. The person who discovered place cells was John O'Keefe. He discovered them in the late 70s, I think, or the early 80s, but I think it was the late 70s. And the people who discovered grid cells and did most of that work on that was a husband and wife pair named Edward and Maybrit Moser. And the Mosers were working, I think, in England at the time, and then they've been running their lab in Norway for a long time now. And so the 3 of them shared that $1 million prize. It's a big deal. And so there's also things called border cells too from the entorhinal cortex. Now let's see if we can take a look at what some of this might look like. So this is an example of a picture of a maze. So you're looking from a bird's eye view down on a maze, and this is where the door where the rat comes in and out of. So the rat is hanging outside and it comes into the maze. Now this is not a very difficult maze, right? You can either go around this way or you can go around this way. Okay? And you can get a little drink of water there, whatever. And what these colors show are different spikings of different cells. So different cells have different electrodes dropped into them and cemented into their head. And those different electrodes are different colors in our map. That is in our graph. So let's say we have orange, yellow, blue, red, green, pink. Huh?
Salmon.
No, there's no fish here. This isn't the water maze. This is like a regular maze, like on the ground.
It was a joke. Yeah, it was a joke towards Erica picking her colors for her wedding.
No, I know, and I had a joke there.
Oh.
So it's mine. So it's just a bad joke. But anyways, but the idea, okay, so it could be salmon or mauve or what have you. So let's say you have 6 or 7 different colors. I'm trying to count out here, right? That means there's 6 or 7 different electrodes dropped into 6 or 7 different cells. And so it shows you that this particular cell only fires when you're in this particular place. This other cell, in orange, only fires when you're in this other particular place. And the yellow firing cell shuts up. Right? And so you can see that's what the colors are useful for showing here. It's showing this idea that cells have a place preference for firing. That is, they remember when they're in a certain geographic space or place. So we call them place cells. So that's how to look at this if you haven't been familiar with these kind of graphs before. And now the other kinds of memories we've already kind of alluded to, right? We talked about sensory memory, we talked about short-term memory. Now the thing about short-term memory is almost nobody really refers to short-term memory anymore in the field. We call it working memory, okay? So just a little context there, that's what most of the literature is going to refer to it as. And that's kind of the idea as the memory that you're holding in mind for about 30 to 60 seconds max, okay? And so it's very short, and it's kind of like, a long time ago, people used to have to remember phone numbers. And now you don't have to because you just type it into your text box, right? And so, yeah, you see what I did with that. But the idea, right, is that a long time ago, people would, you know, if you found somebody cute at the bar, and they'd be like, yo, can I have your number? They're like, yeah. Are you ready? Because now you're going to have to remember it, right? Yeah, or you'd have to go find like a pencil and write it down or something, right? But the good way, so people used to be able to say an example of short-term memory of being like when someone tells you a phone number, you have to hold it in mind. But that example doesn't even work anymore because we don't really do that anymore in society, right? But we used to have to, and that's what a good example of it would be. About 30 seconds holding something in mind, and then it kind of fades away unless you're able to really continue holding it in mind, in which case you're storing it into that long-term memory encoding. Okay? And the sensory stuff is more or less, even before short term, it's just a few seconds of that sensory information, that sight, that sound, that very elementary kind of sort of just sensory information that can kind of be primed later on. So, let's see here. And this kind of shows that temporal comparison about our different kinds of memory, right? So you have essentially that really, really short sensory information that really drags off fast. This is our short-term working memory for about 30 seconds or so, and then it really starts to fade. Now we have two different kinds of long-term memory shown here, right? That all of us are familiar with, right? This is basically what you want to do for this class. This is what I want you to do in this class, right? So we have different goals in that regards, right? I want you to remember stuff. I want to teach for the long term. I want you to remember stuff forever, right? And I understand that you guys just want to know it just enough to sometimes do the test, right? And but right, so, I mean, but the idea here is if you're going to pay for it, you might as well get something that you're going to remember for a long time. And so, but it's an example to underscore that we have kind of two somewhat different models or frameworks for the long-term memory in terms of temporal information. And so, so some of the ways that you can look at short-term memory includes things called a phonological loop, a visual spatial sketchpad, and an episodic buffer. So a phonological loop is kind of like when somebody tells you a phone number that you have to maintain in mind. Phonologically means that you're sort of saying it to yourself in your head. So if I say, remember this phone number, 867-5309. Right? So now you're remembering in your head as you keep going, 867-5309, 867-5309. Right? That's the phonological loop in your head. It's also whose phone number? Jenny's. Yeah, any 80s music fans, there was a big popular song in the 80s that it's actually the name of the song is 867-5309. It's about this girl that he liked. That was her phone number. Yeah, Jenny. And so visual spatial sketchpad, on the other hand, is instead of phonologically repeating that information in our short-term working memory, it's instead kind of holding it in mind visually. So it's sort of like if you have to do like a mental rotation or like visualize something in that regard, it's holding that visual impression in mind in sort of that, sketchpad or draft thing. So different sort of tool for different types of jobs of the different sensory types of information that might be coming in that you're holding in mind. Okay? And the episodic buffer kind of integrates that and kind of works back and forth with our episodic memory to help form some of those longer term memories. And so Alan Badley kind of was the researcher who conceptualized this model. And we can kind of get to that intermediate long-term memory. So now we're looking at this purple side. And that's really sort of object recognition, spatial location, all the kind of stuff we've been talking about for episodic memory. Most of the stuff we test, to be frank. At least certainly in my genre of the research that I think of. We're probably more in this area of object recognition, location, and again, kind of looking at the regions of the brain that are involved heavily, the hippocampus, and again, that much longer term is going to be that cortical areas that we talked about at the beginning of class, having that information exported to. And so, let's see here. When we talk about some of the spatial information, God bless you. Let's see how much, where are we here? We're about 36, 17, okay. Let's see. So this is kind of a nice way to conceptualize that model.